Well, good day, everybody. This is Chris with the Ancient Scholar. I hope this video finds you all doing well. All right, in this particular video, I'm going to continue talking about the so-called current of injury and how it can, at least in some sense, or perhaps partially explain the phenomena of the ST changes that we see that are often associated with ischemia, injury, and infarction. So what I'd like to do is just share a little diagram that I have made here, Notability. So let me share with you all. Here we go. Make this full screen. All right. So if you remember, when we talk about leads, remember a lead is just essentially a voltmeter, and you've got a positive electrode, and you have a negative electrode. And the view, quote unquote, between these two electrodes is what we call a lead. And so when a signal moves toward an electrical signal, like a wave of depolarization in the heart, moves toward the positive electrode, that will be represented as positive deflection on the ECG. And then when a signal moves toward the negative electrode, that'll be represented as negative deflection on the ECG. And when there is no net movement of current, um, that movement of current, that flow of electrically charged ions and anions, cations and anions, um, when there's no net, and that's the key point there. So you can have flow in one direction, another direction, but if it cancels out and there's no net signal moving toward the negative or positive electrode of a lead, then you will get a flat isoelectric line. All right, so that's kind of the basic motivation there. So when we look at the regular PQRST complex, you're looking at the P wave in the P to R interval, which is the SA node firing, the atria depolarizing. That's held at the AV junction for a small amount of time, allowing the atria to contract and preload the ventricles. And then that signal goes through the bundle branches, depolarizes the septum and the ventricles, and you get the QRS complex. And then it should return back to the isoelectric line after the ventricles have depolarized. And there is essentially a plateau when you look at ventricular depolarization, right? So if I look at the action potential here, looks something like this, right? So this flat line here is the resting potential. That's prior to ventricular depolarization. That's what we call phase 4. And then in phase 0, sodium rushes in to the myocytes, and you have voltage-gated potassium channels that then open up here in phase 1, and potassium begins to leave the cell. And so the potential dips down, and then in the plateau or phase 2, calcium ions begin to enter the myocyte, and that offsets the potassium leaving, and you get this flattening out. And so the net, right, the net flow is kind of canceled out. And that's kind of what you get right here at the isoelectric line after the main QRS complex, right? Um, you get kind of this flattening here before you go into repolarization or phase 3, where lots of potassium continues to leave the cell and repolarizes it. But this flattening here is essentially you're having no net flow. You kind of get potassium and calcium kind of canceling each other out, and you should have a return to the isoelectric line here. All right. And then you've got the T wave, which is repolarization. And then you're back into phase 4, the so-called resting potential. So what is going on when we have ischemia, injury, and infarction? So the first thing I'm going to talk about is I'm going to talk about full thickness injury to the myocardium, right? So you have transmural ischemia and injury that involves the entire thickness of the myocardium. So normally, and maybe I can select a a different color. So normally when everything is functioning and the heart is well perfused, your signal is moving from the SA node out through the atria, down the bundle branches, and on average, you have a signal that's moving kind of like this in this direction, right? Um, and so you're going to get, your classic upright P QRST kind of morphology there. Make that go away. Okay. But let's talk about what happens when I have transmural ischemia and injury. So I have the entire thickness of the myocardium involved. And those cells in the myocardium, those myocytes, are going to be ATP depleted, right? Because they're not able to get oxygen, they're not able to undergo sufficient amounts of aerobic respiration, and anaerobic respiration, glycolysis mainly, is not going to produce sufficient amounts of ATP. And there just happens to be a special type of potassium channel called the ATPK channel. And when ATP is present, that channel is closed. And in situations where there is decreased ATP, or rather increased ADP, so dephosphorylated ATP, this potassium channel opens, and that causes potassium ions to rush out of these myocytes in this area. All right. And that is going to cause lots of different changes. You're going to have potassium ions. You're going to have a lot of other things. This is going to be acidotic. So you're going to have hydrogen ions, and eventually it's going to impact sodium channels. You got a lot of complex things, but one of the big things you have is a lot of this potassium leaking out. And so you kind of get this local tissue hyperkalemia. You get kind of really hyperkalemic in this area, and what you get is the resting potential of these cells changes, uh, and the threshold potential changes, and so these cells will depolarize and repolarize abnormally. And what you get is you essentially get a difference in charge, right? So you have a lot of potassium out here, and when compared to myocytes that are next to that area, so these are the healthy myocytes, say, or well perfused, there is a difference, right? Because normally most of the potassium stays in the cells. And so there is essentially a difference in charge concentration, and that sets us up for a difference in voltage, essentially. And it creates a kind of current, if you will, right? So these ischemic and injured tissues essentially are producing a current that is pushing away, it's moving out, right, away from these damaged, dysfunctional myocytes. And the only direction that current can go is toward healthier areas of the heart. And so that produces a quote unquote current moving out in this direction here. All right. And if you look at that, that current is moving, generally speaking, away from the positive electrode, right? And so what you get is you kind of get, because remember your baseline is the isoelectric line, is where you don't have any net flow. But the problem is I have this constant current, this constant difference in charge, right? This constant potential difference, pushing out away from the positive electrode in this direction. And what the thinking is that the isoelectric line gets reset, if you will, right? I've got a lot more, I've got a big charge difference um, in these cells. in these injured cells as compared to these normal cells, and like the threshold potential at which the fast sodium channels open up is changed. And essentially what happens is the isoelectric line kind of gets reset, if you will. The isoelectric line gets shifted downwards, right? So at rest, right, there is a big difference, and the isoelectric line is shifted downward because we have this constant flow, if you will, from these injured and ischemic tissues pushing out in this direction. And so what happens is the isoelectric line gets shifted down, it gets reset, and then you've got the P wave, you've got the QRS, and then you have the T wave. And during the T wave, everything depolarizes, and that charge difference between these that potential voltage difference, really, it's difference in charge concentration between these injured and ischemic cells and these healthy myocytes, oh, in the surrounding tissues. I wonder why it's doing that. Sorry about that. There we go. So that charge difference is kind of reset when everything depolarizes. And so what happens is during the flat line, right, this section right here, the J point actually gets shifted back up to where it would normally be if there was no charge difference, right? Because think of depolarizing as kind of temporarily resetting things. And so what happens during that ST segment is the actual isoelectric line returns temporarily because everything's depolarized and kind of reset. And then we go right back into it. We go back into resting potential, and then you have that constant flow of the current of injury that then shifts the isoelectric line back down. So really what this ST elevation is the return, right? So the ST elevation is really the return of the isoelectric line. Let's see if I can draw this a little better for you guys. It is a return of the isoelectric line to kind of where its normal baseline would be. There we go. So this right here is where the isoelectric line would be if you didn't have this constant current, right? Shifting everything else down. And that's what we think is creating the elevation, or that's at least one hypothesis that explains ST elevation. If you really think about it in some depth, though, you can start to see some of the flaws in this. And so I would say that this is a necessary but insufficient explanation. There has been some research done mainly in the 1990s on animals where dogs, I think, were lesioned. We actually lesioned coronary arteries in dogs, and we gave a group of dogs a chemical that actually blocks these ATP potassium channels. And that actually showed that the amount of ST elevation was decreased in those dogs. So they'd a lesion, or they'd occlude a coronary artery in the dogs, and they would look at dogs that had normally functioning ATP channels, and they would have the classic ST elevation. And then the dogs that had been treated with this chemical that blocked the ATP potassium channel had far less ST elevation. So this explanation does seem to be necessary, but it's certainly insufficient. And I think it's a lot more complicated. There's a lot more going on. And at the end of the day, it's not completely elucidated by any means, at least as far as I can tell. But at least this is a bit of this goes a little ways toward explaining what we see, at least in some cases. And it also helps kind of explain why we might expect to see T wave and ST changes in patients who are hyperkalemic. So in hyperkalemia, you don't have this kind of this localized hyperkalemia you see in transmural ischemia, injury, infarction. You see more of a systemic uh, hyperkalemia within the blood plasma that's surrounding all of the myocytes, but you're still getting these depolarization anomalies that can again help to provide some explanatory value to why you can see T wave and ST segment changes in the setting of myocardial ischemia, injury, and infarction. All right, so hopefully you all found this video somewhat helpful. And in the next video, I will try to talk a little bit about ST depression and how this model can have some explanatory value in terms of SD depression and how it has classically been associated with subendocardial ischemia. Although now in the setting of the OMI paradigm, we know that certain types of ST depression are most certainly not necessarily subendocardial ischemia and may also be present in transmural injury, further complicating our ability to fully explain what is going on the ECG. But at least we have some limited explanatory value. All right, everyone. I hope you found this video helpful. Thank you all so much. Take care. Bye bye.